Hi, my name is Jacqueline Rima, and I'm a rising junior majoring in animal bioscience at the University of Delaware. This summer, I worked in dental labs to learn various laboratory techniques and coding skills while studying ovarian follicles in the laying hen. Hens have a singular ovary that consists of an order of follicles. We would collect these follicles from chicken at the University of Delaware and run tests on them like I will describe later in this video. We live in a technological age where solely working in the lab is no longer sufficient. In addition to doing wet lab work, scientists must take their data and analyze it further with computer software to make more connections and hypotheses. So, this summer I learned how to operate R and run code in order to analyze RNA sequencing data. So what is RNA? RNA is a single-stranded nucleic acid present in all living cells. mRNA is a type of RNA that carries genetic information from DNA to the parts of the cell that makes protein. Only some of the genetic information in DNA is transcribed to mRNA, and that specific information gives the protein its function. This is called gene expression. Now, with RNA sequencing, we work backwards to determine what genes are being expressed. Because of this, we can detect changes and differences between cells. We do this with the help of computer softwares like RStudio. R is the language of data science and is used to analyze large amounts of data. Meanwhile, RStudio is a software scientists use to write scripts in R. Before we can sequence RNA, we must first isolate it. We do this by obtaining ovarian follicles from chicken at the University of Delaware. From these ovarian follicles, we can collect different types of cells. This is a 2-day procedure. On day 1, we add trizol to each test tube containing follicle cells and use a polytron to grind up the tissue. Next, we place the test tube in liquid nitrogen to preserve the sample and repeat these steps for the remaining samples. On day 2, we add various chemicals into each of the samples, such as chloroform, isopropanol, and ethanol, while using the centrifuge between each step to separate the RNA from protein or other contaminants. In the end, we would use a nanodrop to determine the purity of the RNA obtained. Lastly, I analyzed RNA sequencing data. This can include making graphs and charts to help visualize the information. I am very grateful that I was able to immerse myself in the field of research this summer. Through UD Envision, I had the privilege of not only envisioning myself as a scientist, but actually becoming one. Thank you.